Welcome to the Science for the Public lecture series. Science for the Public is an organization committed to bringing science information and issues to the general public. Visit our website for our program listings and blog.
Good evening. I'm Yvonne Steff for Science for the Public, and I welcome you to Contemporary Science Issues and Innovations. Tonight, our distinguished guest is John Beckwith, American Cancer Society Research Professor of Microbiology and Molecular Genetics at Harvard Medical School. Dr. Beckwith's lab was the first to isolate and clone a gene from a bacterial chromosome. This was in 1969, and it was a crucial breakthrough in genetic science, introducing, for example, the potential to control genes associated with inherited diseases. However, the Beckwith group realized that there were also darker possibilities in gene manipulation, and they expressed that concern directly. At his 1969 press conference announcing the isolation of the gene, Dr. Beckwith warned that scientific and public vigilance would be necessary to prevent the negative potential of genetic engineering. And since that time, he's been an unrelenting voice for ethics in genetic engineering. The isolation of the gene at the Beckwith lab was followed with a number of other major accomplishments, including the discoveries of the switches that turn genes on and off, protein folding in cells, and the mechanisms of protein transport both inside the cell and going out of the cells. Throughout his distinguished career, Dr. Beckwith has coupled his scientific work with social activism aimed at raising awareness of the potential misuse of genetic research. For decades, he's been a vocal critic of reductionism in genetics, that is, the tendency to ascribe to an individual's genes complex characteristics such as intelligence and behaviors such as criminality. We'll hear about some surprising examples of this problem tonight. Dr. Beckwith was a member of the Ethics Committee of the Human Genome Project in the 1980s, and since 1983, he's taught a course called Social Issues in Biology at Harvard University, one of the first of its kind. Today, he's a member of the Genetic Screening Study Group, which he'll tell us about as well. Dr. Beckwith has received many awards for his work, including the Abbott ASM Lifetime Achievement Award from the American Society for Microbiology and the Selman Waxman Award in Microbiology from the National Academy of Sciences. He's a fellow of the American Academy of Arts and Sciences also. It is a very great honor to welcome Dr. John Beckwith. Welcome, sir. And we'll start in, if you will, with giving us some background, how you got into this field in the first place, which was odd.
I think in my early days of my career, I was basically carried on by what I did best in my classes. Mathematics, chemistry, etc. But at the same time, when I was an undergraduate in college, I took many courses in literature and other fields other than science. And I ended up graduating having mixed feelings about continuing in science, but I basically let myself be carried on to what the normal path to follow was. And I got into graduate school in chemistry, and I started to get even more concerned about whether this was what I really wanted to do. But during graduate school, I discovered the work of some geneticists, which I knew nothing about beforehand because I had never taken a genetics course, which really blew me away. I was really overwhelmed with it. They were French scientists who had not only done amazing work with very creative, almost magical kind of findings, but at the same time, they wrote very beautifully, and it just carried me along into my career, which I hadn't necessarily expected was going to last. So for several years I made an effort and finally succeeded at finding a position in that laboratory in France where the work had been done.
That wasn't Monod.
There were the two French scientists. There was a bigger group, but François Jacob and Jacques Monod, who made basically fundamental discoveries that are the lore of original findings in molecular biology that really opened up the field of biology along with two or three other important findings.
Right. So you made your way into it in that manner. And then how did you end up at Harvard in the medical school?
I was in graduate school and then at Harvard. Then I went to do postdoctoral work in various places. Yeah. And at some point I wasn't even sure I was going to find a job. I was still trying to get into this French laboratory. And at the same time I was finally accepted, I was offered a job at Harvard Medical School because my work became known at that point. So I took the job because I didn't have any other offers. So that's why I ended up there.
On the other hand, now that couldn't have been when you discovered the error in the genetic switches error. So can you tell us about that? Because not many people just starting out come up with something like that.
Well, I was working at with a professor as my first postdoctoral fellowship at Princeton University.
I see.
And he had started a project which was very much based on the French scientists' work and accepting that it was the theories and the ideas were all correct.
Had they gotten the Nobel Prize for this?
No, they didn't get the Nobel Prize.
Excuse me.
Quite a few, maybe 6 or 7 years later.
Okay.
And so I started on the project, and I just got some very peculiar results. It didn't make any sense, and this doesn't fit with my idol's theories. There's something funny here. And I worked along with it until it became clear that in a kind of small aspect of their work, they'd made a significant misinterpretation of their results. So in a way, I didn't find something new. I had found something that disproved something that well-known scientists had done, and that's enough to make one famous.
Oh, I think so, because that was sort of overnight. And as I say, I don't know how many people start out like that, because that was sort of a groundbreaking thing all by itself. But in any case, you went on and you set up a lab at Harvard and immediately got famous again. Can you tell us, please, something about the isolation of the gene? That was such a big thing there in its technical aspects.
Well, it's funny. There's no discussion of isolating genes because nobody had thought of a way to do it, and I don't know how many people were thinking of it. And in fact, when I look back, particularly in preparing for this interview, I realized that we actually had the tools for doing it 4 years before we did it. We didn't realize.
You didn't know you could do it.
We didn't even realize it. It came about. It's complicated how it came about, but we discovered we had these bacterial viruses that carried the lac genes from the bacterial chromosome, which I had done when I was with the French scientists.
That was their thing, the lac.
And took that back to start my lab with. And a few years later, we isolated another one of these viruses carrying these particular genes, and that actually made it easier for us to realize we could do that, make a pure gene.
Right.
But I think it's kind of a question where people weren't even thinking about it because the tools weren't imaginable at that time. But once we had done this, then people came up with better and better techniques, and it's been moving on ever since then. Within 3 years of our isolation of this pure gene, a technique that made it much easier to cut pieces of chromosomes into small pieces that were gene-sized pieces and use them was developed. And then our our method was no longer useful.
Right,
Yeah.
but now, today, you don't even think about this. It's amazing that it was such a big step at
Yeah.
that time. And again, that was at least you announced it in 1969. So it was 1969, and this is sort of a field is still really developing at this
Yeah.
time, right? So it was a different thing. Now you went from there. I mean, you had one thing after another in this magical lab, and you trained a number of very well-known scientists, correct? So what else did you do in there that was...
Actually, in those early days, we actually went from this issue of the mistake that the French co-workers had made,
Yeah.
because they were defining a particular site that was an important site for turning genes on, and they had mistakenly misplaced it in the map of how the gene works. And so we went on to actually get mutations that affected that site in the chromosome, and were able to define it very specifically, and that settled that question.
Right.
After that, I got interested in moving on to more complex problems in biology, and the next major area we went into was so bacteria are cells with cytoplasmic membranes that surround them.
Yeah.
And they often secrete certain proteins out of the cell into the surrounding media, or in bacterial infections, they secrete proteins into human cells or whatever animal they're affecting. And we were very interested in what's the process that the cell uses to move a protein that's being made inside the cell to outside the cell. And again, we used genetic approaches to isolate mutants that cause defects in that process. And this allowed us to specify features of both the proteins and the cell that allowed this to happen.
I see. So, and then with that work, a number of people also became very famous. So this is a big deal in this lab. Now, over the years, in say zooming up to more recent years, is there anything that you are working on or have worked on more recently that is of great interest to you?
Yes.
Good.
It's keeping me very busy now, which I didn't expect. I expected to slow down.
No way.
We were working on protein structures, and particularly how proteins fold.
Yeah.
And there's one particular aspect of protein folding which is important for a lot of proteins, which is they form chemical bonds between amino acids in the protein that are very far away from each other. They're called disulfide bonds.
Right.
And they lend a lot of stability to proteins, particularly ones that are secreted out of the cell, like the problem we were working with earlier. And over the years, we discovered the mechanism whereby these bonds form, and then began to look at different bacterial organisms to find out whether they all did it the same way, or whether there were different enzymes in each organism to make these disulfide bonds. And we found something that surprised us, and that was that they did differ amongst bacteria. And not to go into detail, but out of that we discovered a way to look for antibiotics that would inhibit the process of protein folding, because bacteria secrete a lot of proteins, or have a lot of proteins on their cell surface that are very important for killing people or killing organisms. That is, they make toxins that are stabilized by disulfide bonds. They make adhesins which allow them to attach to human cells, for instance, that are proteins that have disulfide bonds. So if you could stop the bacteria from making disulfide bonds in their proteins, then you would be stopping the bacteria from making things that are very important for them in causing disease. So as a result of that, just in the last sort of 3 or 4 years, we've ended up with some nice genetic approaches that allow us to readily look for antibiotics that inhibit the process. So we're now doing something I never imagined I would do, because I'm a basic scientist.
Exactly.
We do basic
so
research.
You weren't expecting to do.
But I think it's
pharmaceuticals,
actually, I think it's a very good example of how people doing things in not necessarily obscure regions, but things that don't seem to lead to applications can very quickly lead to societally important findings.
Yes. Would this affect, if you would develop antibiotics, you're saying, that would use this mechanism, but would you also have a problem with mutations with the with the antibiotics? Would that cause a problem? Resistance. I'm sorry.
Yeah. That
doesn't eliminate that problem.
I mean, the reason generally it's important that a lot of people are working on antibiotics at the moment is that organisms of bacteria have become very resistant to a lot of the antibiotics that exist. So people are talking about using combinations of antibiotics because then it's harder for them to resist two very different antibiotics. But this one, this is also a different approach in that it doesn't necessarily stop the growth of the bacteria in every case, but it prevents the bacteria from producing these toxic proteins for humans or whatever they're infecting. So they can still grow, and therefore there may not be selection for resistance because you're not necessarily killing them.
They're sort of neutralized.
So it's not our idea, but it's a new approach that people are talking about now. So people are trying to come up with lots of novel approaches for getting
but that's interesting. You would end up in something like this, a bit far removed from what you had started from. But in any case, now I'd like to go, since you were so well known as an activist, and go back to 1969 here when you announced that the isolation of the gene, and it was a very big deal because of something you did at that time in cautioning where this could go. And I'd like to talk about the reaction, why you did that, and the reaction to it.
I think a lot of it was the reason we called this press conference and not only talked about the work we'd done, but also for the second part of the press conference talked about our concerns, reflected the fact that at that time U.S. society and many other countries were in turmoil over, for instance, the Vietnam War, and in the United States other issues, civil rights, etc., demonstrations in the streets all the time, etc. And also that was influencing at least some scientists who were speaking out. It largely began with scientists who were concerned about testing of atomic weapons. And I think that had a big influence on biologists, at least some of the biologists, about where was their science going and what were the potential dangers of it. So it was in that context that we felt it was important, because we felt there was an issue related to the government and the way it was, what kind of science it was funding and what it was using the science for. And in many cases we thought it wasn't for such good purposes.
So what was the reaction to that within the scientific community? So some scientists were behind, you know, certainly in this area, I think, because I think we talked about before that the Union of Concerned Scientists started up then a number, I think maybe Physicians for Social Responsibility may have a number of these groups of professionals that were very worried about where their scientific efforts might lead inadvertently. That was not their aim. But in the academe, when you really got into this, the next move was a little, you know, more activist, not just speaking out like this. Was there a reaction?
I think there was a like a cleavage to some extent between younger scientists who were more immersed in the protests, et cetera, and older scientists. Two reasons. There was a sense of scientists are ivory tower people.
And they certainly were at Harvard.
And they should be free to continue their work without having to worry about other things. That was society's problem, even though they might be doing the work that's causing one of society's problems. Furthermore, I think I clearly got messages from some of the older scientists that it was not a good thing to do because it might make science look bad, and that might lead to cutting back on funding of science.
Yeah.
Which I found very dismaying. That yes, you're right about those things, but you're going to cause problems for the development of science.
Right.
So it's more sort of self-interest issues. But there were a lot of younger scientists who were becoming involved at that time. Before that was the first, really the first thing I ever got involved in that was political, but also was political connected to science. And other students that year of 1969 at MIT held a big strike about some of the kinds of things that MIT was doing with the development of military weapons.
The thing is, we associate this much more with students than with, say, faculty, but in that time there was a lot of faculty upheaval and support for these concerns also, I think, right? I'd like to go back for just a minute here. Your concern about the misuse of genetics in this case was grounded, I think, in a history in the United States and elsewhere in the world about the misuse of inheritance. Can you tell us anything about that? And I'm thinking of the eugenics movement in this country, and also in that period when you're in the 60s and 70s, the overt racism that was scientifically presented at that time. Can you tell us about it?
When we called the press conference, actually, I don't think we were that aware of the history. We were to some extent.
Yeah.
But what happened was, once you get public, you have to explain what you're saying. We were worried, I think, at the time about the possibility of taking genes, human genes, and engineering humans for various negative purposes. But very quickly, I realized I had to know more about what the hell I was talking about. And so I started to read a lot and discovered very quickly the history of the eugenics movement in the United States, which began in the beginning of the 20th century and spread to many countries, including Germany, ultimately.
Right.
But what happened in the United States in the early part of the 20th century was the eugenics movement, which included the support of many scientists at the time who really believed in the theories that were being put out, that there were people with inferior genes and people with superior genes. Obviously, the people who are making a lot of money and richer must have superior genes, and the ones who aren't making so much money don't. And so they were successful at getting a number of different laws passed, like laws against marriage between people of different racial groups, and sterilization of people who had low IQs, which it was argued was genetic, and in an attempt to breed a pure race in the same way that eventually the Nazis made it even much more explicit than in the United States. But it really started in the United States. And some of the laws that were passed, like in 1907, 1908, were laws that were provided as models for what the Germans did, actually decades later, later than it was done in the United States. And most of the quote research, I use quotes because it wasn't good research, in that field of genetics and human behavior was done in the United States and passed on. Again, provided the source for arguments that were made in other countries for eugenics.
Do you think that has anything to do with our immigrant history? Because a lot of that was imposed on these very poor immigrants that were met with IQ tests at Ellis Island and these other entrance points, and they were all considered deficient, right? Would it have anything to do with that?
Sure, it would. I've said the eugenics movement, but the eugenics movement also was supported by research that came out of universities. And one of the people, Herbert Goddard,
Yes.
was a psychologist, did a lot of studies on immigrants coming to the United States, giving them IQ tests when they came, having different
And
languages.
they were illiterate
very
and poor.
And the science itself, if you
Yes,
read about it, was incredible. But and then he argued that something between 70 and 80% of immigrant groups that were coming from Southern and Eastern Europe were idiots.
Yes.
I mean, that was a technical term in those days.
Did he use the term? Oh, how
Morons
about that?
and idiots. I can't remember.
Yes.
But that they were genetically defective in intelligence. And that was part of the argument in 1924 for a law that actually reduced immigration from Southern and Eastern.
And imposed, what would you call, quotas for different groups and stuff.
Right.
Right. But it's...
So what's really important about that to me, and I don't want to forget to say it, is that it's something that is, as scientists, we never learned. I mean, I did take a genetics course in graduate school. I never heard of eugenics.
Right.
And up until, I'd say, probably the 1980s, geneticists in this country had never heard of it. It's become much more well-known, I think, because of a lot of the activism over the years, over these issues.
And maybe with like Jensen and his overt racism, but it was all doctored up with the IQ tests to cover that, brought it back. Maybe people became more aware of it, just what this is going on, and it's still with us. Obviously, it's still with us. I think that you wrote maybe about pellagra also, which was a wonderful example of how this misuse could come about. But that's a disease that people are not familiar with. Can you tell us about that one?
Yeah, I don't remember all the details, but it's a vitamin deficiency.
Right.
And people are pretty sick from it and not very functional. And it was mostly poor people who had the disease. So there was a debate that went on that was the genetics versus environment debate. There was I've forgotten somebody named Goldberger, I forget which side he was on, but one of the scientists argued that it was genetic, and these were genetically defective people.
Right.
And the other argued, with evidence, because he changed people's diet and things improved, that it was dietary. And he lost. The argument that it was genetic won, and it was persisted for decades.
Oh, I didn't realize that.
Maybe until the 1930s. So this is going back to World War I time.
Right.
When
a lot of people would have had a niacin, I think it's niacin deficiency, and this has an effect on cognitive development and cognition, period. And so it's very easy to say, see, they're deficient, and since they're poor.
So it was accepted knowledge in the government and government committees and healthcare workers that considered this problem. And today we know. There's nobody that argues it anymore.
I'm surprised that the myth was sustained for so long. That's a quite amazing thing. Now,
so it was very much an era when genetics, the laws of genetics, were discovered at the very beginning of the 20th century. In a way, it's very much like the era after the human genome was sequenced in 2000. When everyone was talking about genes, explaining everything again.
Yes.
And that was happening back then.
Yeah.
To be fair to the geneticists of the time, eventually, kind of very much too late, they realized, wait a sec, this is going too far. These genetic arguments are wrong, and we don't support them.
Right.
But it was well after all these laws had been passed.
Well, here's one. This is very widespread now. I mean, as you say, everybody's invoking genes for, I don't know, if you have a hangnail, it's because of genes. But the other thing is that you pinned, and I would like to know if it's widespread in the genetic science community, is you tackled things like sociobiology and what's the other one? Evolutionary psychology, some derivative or other. Tell us about that, would you? And then I'd just like to know if other geneticists are worried about this too.
Worried about the...
that it has become very fashionable to use sociobiology, and there's a couple of very famous scientists who invoke this, that we're rather like the ants or something.
So, not actually being in the field of evolutionary biology, which is the basis of sociobiology, I wasn't really aware that it was a growing field. And so the first time I heard of it was opening up the New York Times one morning in 1975 and seeing this front page article saying, we have a new science which explains everything. The science of everything.
Yeah.
And I was kind of stunned and called up a friend of mine who is an evolutionary biologist, Richard Lewonta, and I said, what is this? I mean, I thought we were finished with these kind of silly things. He said, no, I won't say what he called Edward O. Wilson, but he said, no, he's been into that for a long time, making these arguments that evolution has selected for males and females to play very different roles, and they can't function in science or business and things like that. That racism is innate, and on and on and on.
And behaviors, all sorts, criminality. Well, I don't know about criminality in that case. Other people made that argument.
So it was more sort of general, what is human nature was the argument of sociobiology. And so we agreed that there was no really strong basis for this extraordinary front page story in the New York Times. And we got together a group of people, a lot of from different fields: anthropology, biology, paleontology, genetics, etc. And we spent a summer reading the book to find out how does he say this? And we eventually wrote a critique that caused quite a stir because we found it.
Yes. Was that the letter in the New York Review of Books?
New York Review of Books, right. So that story, I mean, it's still sociobiology. Sociobiology itself is the study of animal behavior, for the most part. It's very little direct studies with humans, and then it's transferred what they find in animals to humans.
Right.
But then there are interesting things that came out of it. One of Wilson's former students was a woman, anthropologist, who studied monkey behavior. And she published a book which is kind of extraordinary. It's really quite interesting to read because she looked at the conclusions that were drawn by studies of male primatologists versus female primatologists. And what she concluded was that the males simply ignored the behavior of females, monkeys, of different species of monkeys. And it just showed how the bias is unconsciously, presumably, affected the scientists who were doing this work. And it was just such a strong example of how it's a problem when you're dealing with human social behavior to abstract yourself and be truly objective about what you're observing. Right.
I hope, though, that geneticists like you will get out there and sort of shake things up a little bit, because it's very easy. And there are a couple of new books out. We were talking about one of them just recently. We're back to this thing. First of all, races matter, and there are these distinctions. And I thought we were through with this a long time ago, but it's back. So I hope that the geneticists will get out there and get vocal again. I would like to ask you about the recombinant DNA a little bit too, because that also has that's been a really difficult thing. First, what is it, and why were you worried about it?
When the recombinant DNA, it's another interesting social history of scientists. This is like around 1974, 75. Scientists started to make genes from different places and put them into bacteria. And for example, they were taking genes from cancer viruses and expressing them in the bacterium E. coli. And a researcher was at a conference sort of proudly announcing that he was doing this wonderful work, and now we're going to be able to study cancer, which in a way it is helpful. When a lot of people stood up and said, you shouldn't be doing that without, you may be spreading cancer genes around in ways you don't want to by doing this. And Paul Berg was the scientist whose laboratory was doing this work at Stanford University. And he actually went along eventually and agreed that this was of concern and one should start to look at it more carefully and to spend some time and stop the research until you have really worked this out and have reason to think that good reason to think that it's not going to be dangerous. The thing that concerned me was more related to my 1969 press conference, because now I was worried about the manipulation of human genes and the possibility of manipulating the human genome. And at that point in my life, although I've changed my view of things, I felt that this research just shouldn't be done, because given the history, we've not used genetics in a beneficial way often, and there's some dangers here. I think a lot of the discussion at the time was really about the health hazards, whether you were spreading cancer genes or other kinds of awful genes. And eventually that got kind of settled. There were committees set up at the National Institutes of Health to protect things. But again, it was scientists moving into an area where they didn't think ahead of what was going to happen with what they were doing. And that's changed to some extent. I think the Human Genome Project, when it started in the late 1980s, along with beginning to support scientific research, also put a fair amount of money into examining the potential social health hazards of the research. So that has changed a bit. So I've been very critical of scientists in the past, but there is some degree of progress. Sometimes it's just window dressing when they do things like that, but there's an idea now that you have to anticipate the consequences of your work. For instance, there's a new field, apparently new field, called synthetic biology, which is doing very similar things as recombinant DNA does, but in much grander ways. And they immediately started that science with ethics committees to express concerns about it. So I think all of the activism in the 60s and 70s around all of these issues by younger scientists and people who were politicized by that era, I think has had an influence. Not to say that things are perfect by any means. I think there are still serious problems. I
was just going to say that I think that right now there's a little concern, like certain industries intruding a great deal in a lot of this research, and you start getting pressured because of funding and so forth to deliver some other kind of product that you may initially object to, but that there is that pressure. I guess maybe that's
also the military. Because there's still a lot of military funding. And there's an interesting article in Science or Nature magazine, maybe last year or a couple of years ago, a debate between two scientists about whether it was ethical to take grants from the Department of Defense that were asking people to develop what were called green bombs. You know what a green bomb is? A green bomb is one so bombs leave a lot of pollution around after they've exploded.
And this is going to make the
So they were going to make the
better.
bombs better bombs because they wouldn't pollute the
non-polluting bomb.
So that was but the discussion in the article was about issues also of funding in science today, where there's so many scientists today, the budget has really leveled off, if not dropped. And people are going to the defense. And not that all of the research they do is bad. In fact, they do a lot of research on diseases in third world countries because our soldiers may end up there someday.
So I realized I was just thinking that the problem is still out there. The question, the ethics questions that you raised, maybe that people are more conscious, is then the other thing that shifts that maybe is this funding issue, which pressures perhaps young scientists in particular, that they don't have as many options, to say. In any case, I need to cover what you are doing now in terms of your work on communicating science to the public and working with students. Your students, you started probably the first course on developing the sort of ethics consciousness among students. Tell us about that, would you?
It actually started, this is in the early 1980s. Students who had taken my genetics course came to me, and in that genetics course I'd give maybe a session or two discussing the last session of the class might be one on ethical or social issues. And the students came to me and said, we really should be getting much more of that. We'd like to have a whole course, and if you'll help us, we will organize it and lead the sessions. So the students actually led the sessions in the first year or something, but then as time went by, we ended up doing, another professor and I from the department ended up doing all the sessions ourselves. And that now covers a whole range of issues. It goes through the history of the eugenics movement. It goes through the history of the idea of race, and from the 18th century on, and how scientists have given different views on it. We look at contemporary research in these areas because these kinds of things still go on and make a splash. We talk about science in wartime, go over the career of J. Robert Oppenheimer, and that's just a fascinating story of the atomic scientists and the mixed feelings they had about what they were doing. Which were things that influenced me actually when I was pretty young, because Robert Oppenheimer was very famous and well known back, and because of
and he had a real conscience.
Yeah, and the conscience hurt him. I mean, he got removed from the committee that was supervising it because of his concerns about how atomic energy was going to be used. And I think that's an interesting example to see what an example of a scientist who has thought about these issues.
Yes. So the course still runs, correct? And you are also engaged beyond that, but I would like to put on the event page, on your event page, then I'll make sure, if I don't have it there now, the name of the course, but it sounds like one of those courses that should be online for the whole world at this point, because I think a lot of people would really like having this, that not just students in university, but the public at large. The MIT and the Harvard online course thing would be just great. But in any case, you also are engaged with another group in terms of putting out information for people. Can you tell us briefly about that?
Right now, there are a couple of things that are engaging me outside of the
like
laboratory. It's a little bit much, but I've been collaborating with people at Kalamazoo College in Michigan on a website where we've started to collect courses like this from around the country and cataloging them, putting them on the website so that people can access these courses. My problem is finding other scientists who I can find courses that deal with many of the issues I deal with. But overwhelmingly, they're more from sociologists and anthropologists and history of science people. Not that there aren't some from scientists also. So we're developing that as a place, and hopefully, and Kalamazoo has a center called the Center for Social Justice Leadership, and they're interested in working with us to really spread the word more about these kinds of courses and to push for it's being something ideally universally that scientists should be, in addition to their science, in their science courses, they should be getting exposure to what it means to be a scientist in terms of potential social consequences in your science itself or in the field of science.
Because it's a scientific era, right? It's a whole new thing. If there's a way to link to that or to give people that link via the event page, let me know, and I'll make sure that because I think people would be very interested. Tonight we have a group of students from a local college, and I know that they're eager to talk with you and ask them questions and so forth. So I am sorry that I have to end there. I could talk about this all night. It's very interesting. Mm-hmm. I really appreciate your talking with us, your coming here and talking with us. And I'll pull back and let you talk with the people in the audience here. Okay. Thank you, and I'm going to move back.
Hello, Dr. Beckwith.
Hi.
So yes, we have a group of students from Endicott College on the North Shore of Boston, and it's an interesting program that we have. There's a major in biotechnology, and the newest major is in bioengineering. And so this idea of synthetic biology and genetic engineering is right at the forefront of the entire program. And along with that, we have a bioethics course. And, I think it's very important that we have that. And it sounds like you're optimistic compared to what it was like in 1969, that there are these ethics committees that are tied into synthetic biology, for instance, right at the outset. But wouldn't you agree that it's also potentially much more harmful, if you will, that there's a chance, there's a good slippery slope argument that things could be out of control and that there is a need for professionalizing bioethics?
There's a need. So first of all, I may have sounded optimistic, but I very much agree with what you're saying. It's overwhelming how fast things are progressing and how hard it is to keep up with them. At the same time, there's still a sector, a hefty sector of the scientific community that really doesn't believe in I found this on the Human Genome Project, where they talk about we don't need philosophers or other people telling us what to do. We know what to do. And I think that's a big problem in it, that the development of ethics, or whatever you want to call it, or concerns about potential dangers of research, should involve people other than just scientists talking to themselves. And I think that happens too often. So yes, I'm optimistic because it was null when I started in science. There was no discussion at all of these issues, and that's changed a lot. So that's hopefully will expand in some way. Do you want to respond to that, or?
Well, I just know that there's this whole idea of the progress trap. That have you ever seen the documentary Surviving Progress by any chance? They describe a progress trap, and the most simplified version of that is when the caveman figured out how to run a whole pack of mammoths off of a cliff instead of just hunting one at a time. And they found this great solution to find a source of food, but they also wiped out the food at the same time. And this idea of a progress trap could be what we're all sort of in the middle of all the time. I know you've been on the forefront of, challenging that and looking for checks and balances. And I wonder sometimes, even teaching in this program, where things might go and where are the checks and balances. It seems, and typically these things come down to politics, right? So whether the Republicans are in office or the Democrats are in office, there's going to be different ethics boards who are kind of declaring what our values are in the United States. So it's a scary situation, and I wonder what you foresee with people like Craig Venter synthesizing whole organisms and so forth.
I'm not sure I want to take Craig Venter very seriously. There's a lot of PR stuff associated. I don't know if you've read, one of my colleagues at Harvard Medical School has talked about breeding Neanderthals. Have you read about that?
Yeah.
And I'm not sure why he's doing it. I'm not sure he ever wants to do it, but I don't know. To me, the biggest problem today is global warming, and that's where I think the issues you're raising are really important.
Right. On the flip side of the Craig Venter issue is that his argument would be that the whole point of this is not to clone human beings and to use eugenics to design the best babies and so forth, but it's to develop chemical products and drugs and things in yeast or bacteria or something in a very rapid production process that without synthetic biology we couldn't really do. So there's this wonderful potential for bioengineering, but there is also this potential slippery slope argument that people are going to do the wrong thing. And the question is, where are the checks and balances?
There are meetings of this ethics group on synthetic biology. I don't know if what they're deciding is available or not. You haven't seen it?
No, I haven't seen anything like that.
But I know that that's part of it. Ethics sections have become part of professional society meetings. Sometimes they may not be terribly meaningful, and I've never had the time to look at what's happening with synthetic biologists.
Do you think that if people were to to alter human genes in order to give more favorable characteristics, wouldn't that eventually narrow down the differences between people? Would that affect evolution in a way? Make us less likely to be able to adapt to a change?
One of the big problems that people talk about in that case is that it's going to cost money to do this kind of thing, and who will be able to afford it? I guess if the government decided that they would change everyone genetically, it could happen, but I don't think that's going to happen. And what people worry about is that it will, in fact, separate people more and more. If it's a little bit, to me, talking about science fiction, because, for instance, at this point, there's no reason to think one is going to be able to introduce genes into people that will affect cognition, intelligence, et cetera, or a lot of other issues that people talk about. It's hard enough for people. One of the big failures of genetics recently is that all of the arguments that came particularly out of twin studies, which people like Arthur Jensen carried out, which said that, IQ is highly genetic, and they'd actually give a figure, it's 80% genetic. When people have gone to look for genes, basically they haven't found them. They've found very, very rare things that have very small effects and don't at all add up to the kinds of things that have been claimed for twin studies. So I think we're in an era where geneticists are following the work that's been done in twin studies on genetics, which has some very serious flaws, and they're discovering that maybe the genes that supposedly existed for criminality or intelligence, et cetera, simply don't exist. That's not to say that there's no genetic effects on people's behavior, but I think there are blanket kind of conclusions that were made by the field of behavior genetics about the overall intelligence and its genetic basis.
So earlier you were talking about how the Stanford University scientists ended up studying the cancer genes and putting them into E. coli. Do you really think that the cancer genes would end up spreading much faster, or do you think something much worse could have happened?
I never was particularly worried about it because I felt that you're actually probably causing a problem for the bacteria. They probably won't grow as well when they have extra genes in them from other organisms. Nevertheless, people did worry that somehow these experiments were always done with the bacterium E. coli, which is a big inhabitant of the human gut. So people in general have E. coli in them and could be infected with an E. coli, which becomes part of their gut. It's possible that the cancer genes could be released and somehow integrated into a chromosome and affect the potential for cancer in individuals. I didn't think the scenario was very likely. Was that the question you were asking? But it was worth pursuing to make, first of all, make people feel better about it. Now this kind of research has been going on for decades, and there's no indication that anything like that has ever happened with any of the bacteria that have been made. And there are regulations developed that allowed one to pursue the research if it was done under very strict conditions where the bacteria was unlikely to escape.
That is, has the genetic base of disease been, is it getting out of scale here so that there's a gene for every imaginable thing? Even in cancer, they're looking, you know, a specific gene causes something. Is that, has that gotten out of whack?
Well, it's gotten out of whack, but mainly out of twin studies, or studies that are not the contemporary gene hunting studies. So if you look at the literature now, there were a lot of early successes with diseases that have straightforward, clear inheritance, like sickle cell anemia, cystic fibrosis, et cetera, and you'd read all the time all these discoveries. But when they go to a lot more common diseases, they are not finding lots of genes that would explain the diseases as largely genetically based diseases. For the most part, they're saying, well, that we're doing something wrong. They're not saying, well, maybe in fact that environment plays a more important role than we thought, which surprises me. I just think people would, and I think it's partly based on the fact that people doing this genetics with DNA, that molecular biologists don't read the twin studies, they just read their conclusions and take it on faith that the genetic arguments made by these people doing twin studies are good scientists.
We're going to stop at this point. We're about out of time, but when we turn on the lights, you should feel free to go and talk to Dr. Beckwith. You don't have to be shy or anything like that.
I'm shy too, so.
He's shy too, so you can all be shy together. But the lights will be on and you relax, get up and go and talk to him and stuff, okay? Thank you so much. Thank you very much.
Thank you.